Hello everyone, and welcome to a new lecture about microscopy. The aim of this video is to overview the principle of confocal microscopy. In the previous lecture, we have described the mechanism of wide-field fluorescence microscopy, which is a very fast technique as we record the image of the entire volume of the sample in one exposure time of the camera. However, it cannot give a complete description of the 3D structure of the specimen. Moreover, for thick samples, it could give rise to blurred images, and there is no discrimination between the plane of focus and the planes that are out of focus. What instead I will explain you today is how to achieve the so-called optical sectioning capability that aims to retrieve the depth position of the fluorophores and therefore of the connected structure. The result will be a set of images, each of them describing a slice of the sample in depth. From this set of images, called the Z-stack, the computer elaborates the 3D structure of our sample, from which we can extract additional information. I will explain how, with an important microscopy technique as confocal microscopy, we can reach this ability. Let's suppose that we want to have the image of a single fluorescent point within our sample, with this simple schematic. In this case, we will need to focus the light to a single spot at possibly high powers, and that's why we will use a laser at excitation source. The laser beam will be directed and focused onto the sample and will excite the illuminated fluorophores. The emitted light from the point that is placed on the focal plane will be collected from the objective and projected onto the camera, producing a well-focused image of that point. However, as the objective illuminates the sample with a cone of light, it excites not only the fluorophore at the focal plane, but also the fluorophore above and below the focal plane. The emitted light from those fluorophores will be as well collected and sent to the camera, but they will form the image above or below the detector. That means that, at the detector plane, they create larger and blurry spots that will sum up with the light projected from the focal plane, therefore degrading the overall image. To solve this issue, what we can do is to place a pinhole where before was the detector. In this way, only the light coming from the focal point will pass through the slit, while the light coming from out of focus points will be in greater part blocked from the pinhole. A detector, called photomultiplier tube, is placed after the slit and will measure the intensity of the incoming light that, thanks to the pinhole, is the one coming from the focal point. So, with this setup, we can discriminate the depth of our focal point and have a well-resolved image of it by rejecting the autofocus light. However, what we want to do is not the image of one single point, but we want an image of an entire section of the specimen. Therefore, we need to illuminate and register the intensity coming from all the points of the focal plane. To do that, we can introduce the excitation beam with an angle offset respect to the optical axis of the objective. In this way, the objective will transform the angular offset into a lateral translation over the focal plane, allowing the illumination and excitation of a different point of the plane. We can then repeat this operation with various angles, and in this way cover the entire field of view. For every exposure time of the detector, we will record one single point. As the angle applied to the excitation beam maps the position of the excited point in the plane XY, we can later reconstruct the position of each point and therefore create the image of the current section of the sample. After the imaging of a slice, we can physically translate the specimen in the depth direction, which is Z, and start the recording of a new section. The images generated in this way are then forming the Z-stack, and they are mapping the depth information of the sample structures. But how do we practically introduce the angular shift of the excitation beam? As shown in this scheme, thanks to a couple of scanning mirrors, we can modify the direction of the light, both in X and Y. However, now, Also, the emitted light from the sample, which contains the information that we want to record, present the same angular shift as the objectives maps position into angles. As the scanning mirrors are remaining at the same position during the exposure time, they will undo the angle change for the emission light and therefore permit to maintain fixed the subsequent pinhole in the center of the optical axis. During this exposure time, the photomultiplier tube will convert the received light intensity into voltage and then into bits that are stored in the computer. As now we want to detect the intensity of the light coming from a single point, which is selected by the current angle introduced by the scanning mirrors, we don't need a camera, but a photomultiplier tube. The photomultiplier tube working principle, briefly, consists in converting the incoming photon into an electron through what is called the photocathode. This electron is then accelerated through high voltages, and as it hits the electrodes, new charges are released, multiplying their number involving the intensity measure. In this way, the photomultiplier tube can also detect very low light signals. So this process is repeated for each single point scanned by the microscope for the current section of the sample, and then the measured intensity are spatially combined together to form the pixels of the image. Resuming, here I presented the working principle of confocal microscopy. Briefly, thanks to a pinhole, it gets rid of the light coming from autofocus fluorophores, gaining contrast, spatial resolution, and optical sectioning capabilities respect to wide field microscopy. However, this gain comes with some expenses. First, it is a quite slow technique, limiting the temporal resolution, as to build up the image of a single slice of the sample, we have to scan and record point by point. Secondly, even if we are not detecting the autofocus fluorophore, they still emit fluorescence. This could give rise to effects like photobleaching, which is the reduced capability of fluorophore to emit new photons as time passes, due to their overexposure of the excitation light. This can lead to strong limitation in making long-term movies of alive samples. These problems have, however, been attached by other microscopy techniques, as we will see in the next lectures. Thanks for your attention.